Good day to you. Welcome back to topic number 3, leukocytes. I'm Dr. Penny Nansamba, a lecturer in the Department of Biomedical Lab Technology and Molecular Biology, BLT, School of Biosecurity, Biotechnical and Lab Sciences, SBLS, College of Veterinary Medicine, Animal Resources and Biosecurity, COVAB, in Makerere University. This content is part of the module BLT 2120, Hematology and Blood Transfusion Science. So we are looking at topic number 3, leukocytes. And this particular time, we are looking at the subtopic 3.22, counting of white blood cells with the Newber counting chamber. The contents include features of the Newber counting chamber. Number 2, manual total leukocyte count. This teaching video has been divided into 2. The first part, where we look at the features of the Newber counting chamber. And in this second part, we are going to look at the manual total leukocyte count. You're welcome to the manual total leukocyte count. Learning objective 2. By the end of this section, you'll be able to explain the principle and technique underlying the total leukocyte or white blood cell count using the Newber counting chamber. Let's look at the manual leukocyte count. We start with the justification for conducting a total leukocyte count. It gives us a measure of the total white blood cells present in a given volume of blood, irrespective of the type of cell. So we don't mind whether it's lymphocytes, leukocytes, monocytes, eosinophils, basophils, neutrophils. We just want the total white blood cells that we are seeing in this given volume of blood. Secondly, we do a total leukocyte count to improve diagnosis and also other decisions concerning treatment or recovery from disease. The total white blood cell count is the total number of leukocytes in a given volume of blood. It is expressed as thousands of cells per microliter or thousands of cells per liter. In a healthy human being, we get 4.5 to 11 to the power 3 white blood cells per microliter, or 4.5 to 11 to the power 9 white blood cells per liter. Let's look at the justification for conducting a manual leukocyte count. So while we've got automated counters giving us speed and accuracy, we might need sometimes to fall back on the manual leukocyte count methods, which involve the Newber counting chamber. So this is done perhaps when the other specialized methods are not available. You may not have electricity, you may not have the equipment, or you may not have the technical skill or the reagents. Then secondly, we might need specific information about the leukocyte subtypes that we are seeing. This case would go for a manual count. Or we might be identifying immature neutrophils or any atypical lymphocytes for specific counts. So let's start on the manual total count. We start with the dilution of the blood sample to enable easy counting of the leukocytes. The rationale of this procedure for the dilution will be discussed. The underlying principles that we encounter need to be appreciated, and we need to know why particular fluid solutions and equipment are being used. So, to conduct a leukocyte count, the diluting fluid that we are going to use will, number 1, preserve the leukocytes, number 2, lyse the red blood cells, and number 3, stain the nucleus of the white blood cells to make them more visible. The common fluid that we use is Tax solution. This is its composition: glacial acetic acid. It lyses the red blood cells. Gentian violet stains the white blood cells, making them visible. And sodium chloride will preserve the physiological state of the white blood cells. This is brought to volume using distilled water. We use a dilution of 1 in 20, 1 part blood, and 19 parts dilution fluid. Now let us proceed to do the dilution. And in this particular stage, we are using a white blood cell pipette. So we tilt the blood tube as shown to be able to draw the blood, and put in the white blood cell pipette as is seen in that picture, and draw blood up to the 0.5 mark. We remove it from the blood sample, clean the excess blood, and then use the same white blood cell pipette to draw dilution fluid up to the 11 mark. From here, we are going to roll the white blood cell pipette several times horizontally in our palms to allow the white bead to mix the blood and the dilution fluid. We shall also give it 3 minutes to allow for lysis of the red blood cells. Now, mouth pipetting may not be acceptable in many labs, or we may not even have the proper suction pipettes. So we are going to use macro methods to dilute our blood sample. So in this particular slide, we are showing you the dilution methods for a blood sample using an Eppendorf pipette. We are going to draw 190 microliters of the dilution fluid or Tax fluid and put it in a tube. Then with another Eppendorf pipette, draw 10 microliters of well-agitated blood sample and add it to the tube containing our dilution fluid. We agitate the mixture gently and set it aside for 3 minutes to permit lysis of the red blood cells. Now we can move on to charging of the Newber counting chamber. We are going to discuss the procedure and appreciate the errors that can occur. The suspension of blood cells and Tax fluid in either our white blood cell pipette or our test tube is agitated, and then with the tip we charge the counting chamber. If we're using a white blood cell pipette, we discharge the first drops and then charge the chamber. The picture here shows charging of a Newber counting chamber. We are going to take care not to overfill or to underfill the chamber. Still on charging of the Newber counting chamber, we are going to appreciate the errors that can occur. The picture A on the right shows a properly loaded Newber counting chamber. Both chambers on both sides are properly loaded. If we look at the picture on the right, picture B, we see some errors have occurred. At the top, that part of the Nuba counting chamber has been underloaded. We can see some space remaining. At the bottom, that section has been overloaded. Some of the fluid has flown into the moat, overflown into the moat, and we can even see an air bubble. All these are going to lead to errors during the counting of our white blood cells. Now that our Nuba counting chamber is charged, we can view it under the microscope. Just by way of revision, the counting grid looks like that under the magnification of 10 or even 4. We are going to count the cells in the 4 corner squares. Each has got an area of 1 millimeter by 1 millimeter, bringing the total surface of white blood cells counted, used for the white blood cell count, to be 4 times 1 millimeter squared. So we place our Nuba counting chamber on the microscope stage and use the ×4 objective to focus. When we see the grid pattern, we move to the ×10, and now we can even focus on the white blood cell particles. This is one of the 4 corner smaller squares. We can see it has 16 smaller squares therein, 4 by 4. Count all the white blood cells. If you're wondering how they look like, they're these small black dots. We count all of them and also do the same for the remaining 3 corner squares and record our value. Counting should be done in a systematic manner to avoid double counting of cells. We start from the right side of the square, move towards the left, count all the cells in those topmost row 4 smaller squares, and move down, count the second row, moving from the right towards the left, move down, count the cells in the third row from the left to the right, move down to the 4th row, and this is the final row, counting the cells from the right to the left, as illustrated in that diagram. Again, to avoid double counting, we are going to count only the cells touching the left side and the top side. So we will not count the cells touching the right line and the bottom line. Since we are done with counting, we can now go to the final calculation in the enumeration of leukocytes per unit volume. We use the formula given there: particles counted divided by the surface area counted times the depth of the chamber times the dilution. We've got our figure for the cells counted in all the 4 corner squares. The surface counted would be 4 times 1 millimeter squared. The depth was 0.1 millimeters, or 1 out of 10. The dilution is 1 in 20, which is 1 out of 20. We simplify the formula to get cells counted times 10 times 20 divided by 4. And you can simplify it further to get the cells counted times 50. And this gives us the cells per unit volume, which in this case can be per liter. In a healthy human being, you'd get 4.5 to 11 to the 9 white blood cells counted per liter. Now let's apply this in the study question 2. We're going to calculate the absolute white blood cell counts from the differential counts. Let's have an individual who got a white blood cell count of 13.5 times 10 to the 9 per liter. This individual is quite sick. And our question is calculate the white blood cell counts for the different cell types. I'll repeat that. Calculate the absolute white blood cell counts for the different types of leukocytes in this blood sample. For this, we need the differential counts. Our differential counts have been obtained from a different assay. Neutrophils gave us 65%, basophils 1%, eosinophils 2%, monocytes 5%, and lymphocytes 30%. We're going to use this information from the differential counts to calculate the absolute counts for all of these different white blood cell types. So it's simple. We are going to get the differential count multiplied by the total white blood cell count. In this case, it's going to be 65% times 13.5 to the power 9, or 65 out of 100 times 13.5 to the power 9. And it's going to give us 8.7 or 8.8 to the power 9 neutrophils per liter of blood. We can calculate the others for the other leukocyte types, and we find that the absolute counts are more accurate in defining disease situations compared to the differential counts. I would like you to come with me and visualize an actual leukocyte count performed using a Haemocytometer. It's given by the Hematology Lab series, and it's a duration of about 8 minutes. Just watch the first 5 to 6 minutes. The URL link is given in the video. Now let's review what we've just learned. What is the function of the tax fluid? B. Why is the manual white blood cell count chosen over automated methods? Our answers: tax fluid lyses the red blood cells, making it much easier to count the white blood cells. And the presence of immature white blood cells, which we may need to quantify, might be one of the reasons why we are preferring to use manual methods over automated methods. In summary, the principles learned for the total leukocyte counts include the dilution of the blood sample to ease counting with a specified volume of blood, the role of the different components of the dilution fluid, and lastly, calculation of the white blood cells per unit volume. Our next steps would be to refer to the lecture notes given to you in the study portal, the activities therein, and the chapters in the textbooks that we have recommended to you for further reading. These are our source of information. And thank you for watching part 2.